Hello everyone, very good evening guys. I welcome you all to this Biotecnika's YouTube channel. So please confirm if you can hear me and see me. Good evening everyone, welcome back. So today we are going to understand about the phases of plant growth, what all phases are there for the growth of the plant, and along with it we are also going to deal about the phytohormones, that is plant growth regulators. So if you have any queries regarding this series, so today it is the 13th day of this series. If you have any queries, you can join our Telegram group. The link you can find in the on the screen, and if not, you can also mail us at info@biotecnika.org. There you can post your query and technical team will revert you back immediately. Welcome dear tech. So we'll wait for 1 or 2 minutes and then we'll start our session. I also request every one of you to stay tuned and connected till the last part of it. And at the last, you will be getting some freebie or some downloads where you can find the important topics for the chemistry, physics, as well as biology. So please stay tuned and connected till the last part of this. So, okay, so now we'll continue with the session. So firstly, we'll understand what is growth. So what is growth? Growth, it is a characteristic of all living beings. It is a irreversible and permanent change that cannot be reversed further. This is accomplished by the increase in cell size, where the cell increases in size, increases in length, increases in volume, surface area, as well as cell number. So how it is performed? It is accompanied by the metabolic processes that occurs at the expense of the energy. So can we say that growth, it is an active process? Yes, this is an active process. It requires the need of energy in the form of an ATP. So whenever I'm talking about growth, I'm signifying structural differences in the cell. And once the cell achieves all its structural properties, now it is capable of differentiating. What is differentiation? When I'm talking about differentiation, I'm focusing on the functional differences or functional implications of the cells. So once the cell gains all its structures, it is now capable of performing a particular function. That is what called as development. So when I combine growth, that is structural changes in a cell, along with functional implications of a cell, that is what called as development. And then what is growth? Growth is just structural changes in the cell that can be in the form of cell size, length, volume, surface area, as well as cell number. So what are the different phases of growth? So we all know that there are 3 phases of a growth in plant. The first is meristematic phase, the second is elongation phase, and the third is maturation phase. The meristematic cells phase, meristem stands for to divide. So it is a phase of cell division where the cells are characterized by the presence of dense protoplasm. Now what is protoplasm? It is a combination of cytoplasm plus nucleus. When the cytoplasms of the cell that bears a conspicuous or big nucleus, that is what called as protoplasm. And here the cells are highly respiring at this stage, and now they will cause the formation of primary cell wall. Now the plant cell, they are having two types of cell wall. One is primary and the other one is secondary. The primary cell wall formation, that is the initiative of cell wall, it firstly occurs in the meristematic phase, and once it is completed, it gets switched to secondary cell wall formation. That happens in the elongation phase. Now coming to elongation phase, this phase is also called as the phase of enlargement, because here the cell will enlarge. How it will enlarge? Because the cell is dividing and it is forming at the time of meristematic phase. Now once it is formed, it's a time to divide for the cell. It will enlarge and it will begin in increase in size. How it will enlarge? It is due to the formation of secondary cell wall that is formed at the time of elongation phase, and along with it vacuole formation also occurs here. Now, what is the significance of forming this vacuoles? So you all know that in the plant cell, it has one big vacuoles at the center that will occupy the maximum space of the cell, and that is why the cell starts enlarging, because the cytoplasm increases, the vacuole formation takes place at this stage only. Now the third phase is called the maturation phase, that is also called as the phase of differentiation. Now, the cell, if it is attaining all its structural differences with the help of meristematic phase as well as elongation phase, that's the time it will enter the maturation phase, where it will differentiate and gain all its functional differences or implications. Now, there are two types of plant growth rates. What is plant growth rates? When we study the increase in height and length of the plant per unit time, that is what called as plant growth rate. Rate here stands for per unit time. So studying the growth of a plant per unit time, that is what called as plant growth rate. Now it can be of two types. It can be arithmetic growth rate. It can be geometric plant growth rate. Now what is arithmetic plant growth rate? Here the cell, that is a dividing cell. So you can see here this is a dividing cell. It undergoes mitotic, that is mitosis, that is cell division. When it undergoes cell division, it will form the same similar cell as itself. It will form one daughter cell, and at the same time one differentiated cell will be formed. Now this daughter cell will retain the ability to divide and will continue in doing so. Now what is the significance of forming this differentiated cell? Now every time once the daughter cell is formed, a big differentiated cell is also formed that will have a particular and defined function in the plant body. So the cell that is dividing, it is forming daughter cells as well as differentiated cell. But what in case of geometric plant growth rate? Every time the cell that has the capability to divide, it will undergo mitotic division, as in case of arithmetic division. It will undergo division, but this time you will get two daughter cells. Here it was forming one daughter cell as well as one differentiated cell. But here you will get both the daughter cell that will have the retain the capacity to divide, and they will continue in doing so. Now here the cell divides with the formula of 2 to the power n. This is the formula for exponential growth rate. So geometric growth rate, it is also called as exponential phase, where the cell will divide with the power of 2 to the power n. Now what is n here? N represents the number of divisions. So now let's see how this is taking place. So if I plot the graph for the arithmetic growth, we'll get the linear curve. What is this? This is the linear curve. Why we are getting the linear curve? Because when we are measuring the height of the plant with time, we are taking height of the plant on a y-axis as well as the time, that is a variable factor on the x-axis. Whenever we are plotting the graph, as the time increases, the height of the plant also increases. There is an increase in the height of the plant with time. That means if we draw the plot a graph, we'll get a linear curve. That is why we are getting the linear curve. That means the time is directly proportional to the height of the plant. As the time increases, the height or length of the plant also increases. But if the time remains constant, the height of the plant also remains constant. So if we signify or if we represent this growth rate, arithmetic growth rate in the form of a mathematical representation. So it will be like length of the plant or height of the plant at time t is equals to height of the plant at time 0 plus growth rate of a plant per unit time. This is what the equation of an arithmetic growth rate. So many a times the examiner just asks you for the equation of arithmetic growth rate. So they will give you 4 options, and out of this you have to select one option for arithmetic growth rate. Then what is exponential or geometric growth rate? Geometric growth rate, as I already stated, this is also called as exponential growth rate, because here the cell exponentially divide in the form of 2 to the power n. What is this 2 to the power n? Suppose one cell is there, it will form 2 cells. 2 cells are there, it will form 4 cells. 4 cells are there, it will form 2 cells. So 2, 2, 2. That means it is dividing in the form of 2 to the power n. That is, n number of cells will be formed. Exponentially it is dividing. So when we plot the height of or weight of the organ of a plant with respect to time, that is again on the x-axis, because variable density of vector we are taking always on the x-axis, and the constant one, it is always on the y-axis. So when we plot, we'll get the sigmoidal curve here. What is the sigmoidal curve? We'll also call it as S-shaped curve. Now this S-shaped curve, it has 3 phases. The first phase is called the lag phase. The second phase is called the exponential phase. The third phase is the stationary phase. What is the significance of lag phase? Here the cells of the plant adapt to the environment. They just read the environment and start adapting. And after it completes its lag phase, it will enter to the exponential phase. This is what the cell division phase. Actual cell division takes place at the time of cell exponential phase. So lag phase is also called as a cell formative phase, because cells are formed here, they are adaptive to the environment. Once they are adaptive to the environment, that's the time they will enter the exponential phase, where they will start dividing. And they will dividing, kaise they will divide? Exponentially, that is 2 to the power n. So it is the maximum and highly dividing phase, and lag phase where you will find the slow cell division. Now this exponential phase, where you can see the highly dividing cell division rate, that is occurring more, and it is followed by stationary phase. Once the cell is fully divided, it will reach to the differentiated cell, it will mature and stop dividing at some point of time. This is what called as stationary phase. As you can see the dip in the graph, here it is constant, where the cell will differentiate, will become mature and stop dividing after some point of time. So if I express this geometrical growth in the form of a mathematical representation as like arithmetic growth, I will get length of the plant at time t is equals to length of the plant at time 0 is equals to the natural log, that is a base of natural logarithm, rate of growth of a plant per unit time. Here E represents the exponential growth. That is why we are getting the sigmoidal curve or S-shaped curve. So E represents the base of the natural logarithm. This is natural log, and natural log, that means you will always get the S-shaped curve of the sigmoidal curve. I hope this is clear to everyone. So if you want to quantitatively compare the growth of the plant, you can do it by 2 methods. One is called the relative growth rate, and the other is called the absolute growth rate. Now, what is the absolute growth rate? Where you are not comparing anything. You are just focusing on the growth of a plant per unit time. Where you are not comparing anything, you are just focusing on a growth of a plant per unit time. That is what called as absolute growth rate. How we can see here? So you can see a leaf A is given here and the leaf A dash. This leaf is expanding and it is gaining its size and it's becoming A dash. Firstly, it was having a surface area of 5 centimeters square, but after some time it has become 10 centimeters square. So you can just minus final value to the subtract to the initial value, and what you will get 5 centimeters square. So this is what called as absolute growth rate. You are just investigating or determining or examining the growth of a rate after some point of time कि ये लीफ ए जो था ये ए डैश कब तक बना, कितने पॉइंट ऑफ टाइम में. And when you are comparing this 5 centimeters square with time, that is what called as absolute growth rate. But what in case of relative growth rate? We are finding the growth of a plant per unit time, but here we are considering the initial value. We are considering the initial value and then we are calculating the relative growth of a plant. How we are doing so? Like in case of absolute, we subtracted the final and the initial value. Here also we'll do the same. 55 minus 50 is what? 5 centimeter square. And here we have to compare it with the initial value. But here we were not doing any comparison. We were just subtracting the final minus initial value and then we were signifying it with the help of a time. We were taking it with the help of a time. But here, when we are subtracting this value, we have to also consider the initial value, that is 50 centimeters square. So we will divide it with the help of 50 centimeters square, and here we have to find the percentage into 100. And then when we are doing all this calculation, we are just combining with the initial value, that is what called as relative growth rate. Relative means related to something, related to initial value. When it is compared with the initial value, that is what called as relative growth rate. Now what you will get? 50 and 2. 10% of growth is achieved in this leaf. As you can see, 10% of the growth is achieved and it is becoming B to B dash. So when we are comparing the initial value, that is what called as relative growth rate. And when we are not comparing any initial value, we are just studying the growth of a plant with respect to time. That is what called as absolute growth rate. Now we will understand what are the sequences of the developmental process in a plant cell. So firstly, a meristematic cell will there that will have the capability to divide. It will divide and divide. And after one time will come, it will attain the increase in the cytoplasm. The cytoplasm content in a cell will increase. That will lead to the expansion of the cell. The increase in the cytoplasmic content of a cell is called as plasmatic growth. Why plasmatic growth? Because the plasm is increased. What plasm? Cytoplasm is increased. So it is called as plasmatic growth. Once the cytoplasm is increased, it will expand. Now you can see an overlap between the plasmatic growth as well as the expansion rate. Now once the cytoplasm will increase, definitely it will expand. Now here you can see the overlapping in expansion rate. Now once it expands, it will attain the property of differentiation. Now all the structures of the cell have been made. Now once the structures of the cell has been attained, it's time to undergo differentiation where it will have the functional implications. It will acquire its functional space and now it will be committed for a particular function. It will have its particular function in the plant body. And after differentiation state, now it has become fully matured. Now this mature cell will not divide further. That's why you're not seeing any kind of arrow here, because this is the last stage of the cell where the cell stops dividing. That's the time the cell enters the senescence and aging. What is senescence and aging? This is a programmed cell death, where the death of the cell will occur. At some point of time, the cell will die. But what happens? This whole phenomena, it is called as differentiation. This is called as differentiation. But sometimes there is an adverse condition in the environment where the mature cell will enter again into the cell cycle. It will enter again to the Z0 stage, that is the meristematic stage, and then it will again start its life cycle. This is what called as de-differentiation. D means going back, differentiation. It has already attained its differentiation stage or differentiated stage. Now it is going back to the original position. This is what called as de-differentiation. And again, when the cell has already reached a de-differentiated stage, and when it will find the suitable condition for it, it will again undergo the cell cycle. And again, it will start the same process, that is what called as re-differentiation, because it is re matlab again, it is again starting for the differentiation process So it is called as redifferentiation. So we have studied three phenomena in which the cell is undergoing growth. It is attaining a differentiated state. That is what called as differentiation. And when the adverse condition is there in the environment, this matured cell, it will again enter into the G0 stage of a cell cycle and will become a meristematic cell, where it will again redivide and again will gain differentiation. Going back from the mature cell to the meristematic cell and entering again into the cell cycle is called as de-differentiation. But once de-differentiation is completed, now cells will again differentiate and perform all its function and it was doing in the differentiation phase or in this diagram. So this is what called as redifferentiation. So now we'll undergo with plant growth regulators. You can also call it as plant hormones and phytohormones. So firstly, we'll undergo with the discovery of plant hormones. So there are mainly 5 hormones that you have to study that they're in NCERT book. First is Auxin, second is Gibberellin, third is cytokinins, fourth is ABS stands for Abscisic Acid, and fifth is Ethylene. So the discoverers for the Auxin, the credit it goes to F.W. Wendt. He is the only person that talked about the Auxin. But how this discovery had happened? So firstly, this Auxin, it was studied by Charles and Francis Darwin. He took canary grass for his study and he explained the phenomena of phototropism. What is phototropism? Photo stands for light here and tropism means movement. Suppose this is a plant. When it is directly exposed to light or to sun, it will cause the release of a substance, of a chemical substance at the tip of the plant. That is what called as shoot apical meristome. It will cause the release of a chemical substance at the tip. And when this substance get accumulated, the cells in the shady part, it will start dividing. So suppose this is a shady part of the cell where the part of the plant is not directly exposed to light. So now this is the shady plant. All the cells, all the chemical compound, they will accumulate here. Now, once they will accumulate here, they will have the highly dividing capacity and division rate at this point only. So what will happen next? The cell that has got the chemical substance here will bend towards the light. Because the chemical substance is here, it is causing the high division rate, which will cause the plant to bend towards the light. So here occurs the cell division phase. So Charles and Francis Darwin, they only explain about the phenomena of phototropism. But after some point of time, this was proved by F.W. Wendt, and F.W. Wendt called this chemical substance as Auxin. That is why the credit of the discovery for this Auxin, it goes to F.W. Wendt, not Charles Darwin. And the material that F.W. Wendt choose for his study is Avena ka coleoptile. What is Avena? Oat coleoptile. But Charles Darwin, what they used for his study, they used canary grass, but they only explained the phenomena of phototropism. But F.W. Wendt, he proved that this Auxin is a chemical substance that is causing the phenomena of phototropism or the bending of a plant towards the light. So now coming to Gibberellin. Gibberellin, the credit goes to Yabuta and Smoky because they are the only one who isolated the gibberellin or the gibberellic acid. This is an acidic compound who isolated the gibberellin from the fungus. And the fungus which is used for the gibberellic acid or for its production is called as Gibberella fusicorae. This fungus, it is known to be in association with the rice seedling. So what happens? Suppose this is a rice plant and in its root there would be a fungus. What fungus? Gibberella fusicorae. It is in association with the roots of the rice plant. So what it will cause? It will cause the height of the plant to increase. The height of the plant will increase and they will be pale green in color. And after some point of time, these plants, after attaining a maximum height, they will fall down. Why they will fall down? Because they are sterile. They are not fertile. They are not capable of producing any progeny in the next generation. That is why they are sterile and they will fall down. That is why this seedling, it is also known as foolish seedling because they are not capable of producing progeny in the next generation. So this gibberellic acid, it was isolated by Yabuta and Smoky. So the credit goes to these scientists. What about cytokinins? So cytokinins, it goes to Skoog and Miller. So Skoog and Miller, they both were preparing media for some kind of plant growth in the lab. But there they also had herring sperm DNA. Herring fish ka sperm DNA. So they autoclave this sperm and put it in the plant media, what they were preparing. What did they see? After some time, the growth of the plant was enhanced. They were getting good shoots in the plant growth. Now, what they did, they isolated one compound from this plant, and this compound is known as kinating. This is the very first cytokinin that was discovered by Skoog and Miller. But this cytokinin, it was not the plant-based. It was the non-plant-based cytokinin. So the very first cytokinin that was a plant-based, it was isolated from corn, that is zeamase. Now this cytokinin is known as zeatin. So zeatin and kinating, they are the examples of cytokinins. And for ABA, that is abscisic acid, no one is known for this discovery because there were a lot many scientists that were working for abscisic acid, and they called this hormone as abscisic acid as well as doming. That is why the discovery of abscisic acid, it is not going to credit, it is not going to anyone. And for ethylene, it is going to cousins. Cousins ye kya hai? It is name, not brothers wala cousin. It is going to cousins. So what are the precursors for this hormone? Very important side. You should mark a 3 mark for this, 3-star mark. So for auxin, it is a tryptophan. What is a tryptophan? It is an aromatic amino acid. So the precursor for this auxin, how the auxin is formed? It is always formed from the initiator or the precursor, that is what called as tryptophan. It is an amino acid. And for gibberellin, it is acetyl-CoA. You all know that after glycolysis, you are getting a glycolysis, which is a one step in respiration, you are getting acetyl-CoA. Firstly, this pyruvic acid, it is oxidized to acetyl-CoA. So we can say that acetyl-CoA is a connecting link between glycolysis and Krebs cycle. Now this acetyl-CoA is only responsible for the formation of gibberellic acid or gibberellin. What about cytokinins? They are formed from modified purines. Important. What are purines? We have two kinds of bases in DNA. One is purine and the other one is pyrimidine. Under purine, we have two categories. One is adenine and one is guanine. So when adenine and guanine, they both are purines. When these are modified, they will form cytokinin. So how cytokinins are formed? They are initiated by a precursor called as modified purine, that is modified adenine, or it can be modified guanine. What about abscisic acid? Abscisic acid is formed from xanthine. It is a kind of pigment, or you can say phenolic, which is a secondary metabolite that is produced by the plant. So abscisic acid ka precursor, it is xanthine. And what about ethylene? Ethylene is a gaseous hormone which is formed from an amino acid called as methionine. What is methionine? It is a sulfur containing amino acid. So I hope this slide is clear to everyone. For auxin, it is tryptophan, which is an amino acid. For gibberellin, it is an acetyl-CoA. You can learn in this way, like gibberellin, firstly glycolysis is happening. This will form pyruvate, and this pyruvate will be converted or oxidized to acetyl-CoA. That is why acetyl-CoA glycolysis means G, and you can recall this way, G for gibberellin. So acetyl-CoA is gibberellin. For cytokinin, there will be a modified purines. For abscisic acid, you will have a pigment that is produced by the plant itself, that is xanthine, and for ethylene, it is methionine, that is again an amino acid. So we'll now understand what are the bioacids that you can perform for these kinds of phytohormones. For auxin, it is an Avena curvature test. What is Avena curvature test? That I've already explained in the previous slide. Avena stands for oat. That was explained by F.W. Wendt. When the plant, it was directly exposed to sun, so it was causing the release of some chemical substance that were accumulated at the shady parts of the plant. And this chemical substance, it was causing the plant cell to grow towards the lighter side, or towards the where the light is there. So after some point of time, the plant will bend here, and this is the bioassay for Auxin. That is what called as Avena curvature test, because Avena oat, it is curving towards the sun side, or where there is more light. For Gibberellin, it is the synthesis of alpha amylase and proteases in germinating grains on cereal. What is alpha amylase? So alpha amylase is an enzyme which is known to break the amylose, that is the branching unit of the starch. So starch, it is formed by amylose plus amylopectin. So suppose if a germinating seed is there and we are putting water, it is soaked in water. What will happen? It will start germinating. You will have seedling after some time. Why this is happening? Because the seed is absorbing water from the surrounding, that is causing the respiration. And this respiration, at some point of time, alpha amylase enzyme will be produced that will break all the starch molecules that is there in the seed. Now starch, we all know that it is composed of amylose and amylopectin. So this amylase will target on the amylose that is making the starch granules, and after some point of time, this amylose, it will be broken, and we will get the energy. And this energy, it will be used by the seeds and it will grow, and that's why we are getting the seedling. So how the seeds are germinated? It is because of the alpha amylase enzyme that is acting on the starch or the stored food in the seed and causing the seedling to grow. That is what called as the alpha amylase production. Who is doing this? Gibberellin is helping in doing so. That is what, this is the bioassay for Gibberellin. That is the alpha amylase and proteases in germinating seeds, grains on cereals. Now what about cytokinin, cell division test? Because cyto stands for cell, kinin means division. So you can clearly see that this hormone is known for the cell division. So the bioassay for this hormone, it would be cytokinin or cell division test. It is also known to preserve the chlorophyll from degradation. That is why this chlorophyll preservation test, this is again one of the bioassay for cytokinin. And for abscisic acid, it is known to antagonist to GA. Whatever GA is doing, it is doing the opposite of GA. So suppose GA is helping the germination of seed. So this abscisic acid, it will inhibit the alpha amylase enzyme and it will not cause the germination of seed. So can we say that it is an antagonist to GA? Yes, it is an antagonist to gibberellic acid, GA or gibberellin. So it will inhibit the inhibition or synthesis of alpha amylase enzyme in barley endosperm test, that will not cause the alpha amylose to produce. It will not act on amylose, that is a part of starch, and no energy will be produced. If no energy is there, it will not be used by the seeds and seeds will not grow at all. And for ethylene, we are knowing the triple response test. Whenever there is an ethylene in the environment, or suppose if I'm making some plant and here I'm providing it ethylene in the media, what will happen? It will cause the roots of the plant to grow. But roots of the plant, they will grow, they will be very short. The roots of the plant will be very short. They will be fat. The roots will be short. It will be fat. And also there happens a lateral growth. How lateral growth? It will not cause the plant to grow like this. The plant height will not be so much, but it will cause the plant for the horizontal growth. Plant will only grow laterally. That means it will cause the lateral growth of the plant or sideways growth of the plant. That is what called as triple response. It will cause the formation of short root. That root will be fat and only lateral or sideways growth you will be seeing. This is what called as triple response. Three response you are seeing when you are providing ethylene to the growth media. This is what called as the bioassay for the ethylene hormone. Now, what are the types of hormones? So we all know that for Auxin, there are different kinds of natural as well as synthetic hormone. When I'm focusing on natural, that are of a natural origin. So what we have under natural origin, IAA and IBA. What is IAA? IAA is the indole acetic acid. This is the most extensively used auxin for the plant growth culture. Now, auxin, it is used for root initiation. So IEA, can we say that it is used to initiate the growth of the roots in the plant? Yes, it is used for the growth of the roots in the plant body. So for this, we provide IEA, that is indole acetic acid in the growth medium. And the other one is IBA, that is indole butyric acid. Here A stands for acetic and here B stands for butyric. So indole butyric acid. So these both are the auxin that are of natural origin. So we do have synthetic hormones for auxins. So what are the examples of these? It is naphthalene acetic acid, that is NAA24D 245T. So these are the examples of synthetic auxin, and out of this 2,4-D is highly used in agricultural feed and it is used as a weedicide. What are weeds? Weeds are unwanted plants that grow along with plants. So suppose this is an field for a barley plant. So along with it, some kind of unwanted grow, if it is growing here, that is what called as weed. So it will take all the nutrients for this plant and this will be consumed by weeds. So that will deteriorate the growth of the main plant. This is what called as weeds. So here we are applying 2,4-D, which is acting as a weedicide. So if you want to apply weedicide to the agricultural field, you can just spray 2,4-D hormone, which is a synthetic hormone or synthetic auxin. So gibberellin, for gibberellin we have 100 different types of gibberellin, but the most extensively used gibberellin is GA3. This is the most used gibberellin which are using for plant tissue purpose. For cytokinins, we have three kinds of cytokinin. First is a kinetin that was isolated and discovered by Skoog and Miller, but this is a non-plant based product. Zeatin, it is a plant-based cytokinin that was isolated by Skoog and Miller from Zea maize, that is corn. That is why it is called as zeatin. Cytokinin that is isolated from Zea maize, and when we are combining it is called as zeatin. And also one is 6-benzyl aminopurine. I already told that for cytokinin the precursor is purine. For cytokinin the precursor is purine. So you can just recall like this: 6-benzyl aminopurine, that is a kind of or example of cytokinin. What is abscisic acid? What is abscisic acid? So for the examples of abscisic acid, it is ABA and also dormin. So dormin, abscisic acid is also known as dormin. Why dormin? Because it is dorminating and it is helping in seed and bud dormancy. It will cause all the seed to enter to the dormant stage. Why it is causing to enter to the dormant stage? Because it is inhibiting the production of alpha amylase, as we have already seen in the previous slide. That was initiated by gibberellic acid, but this abscisic acid, it is cutting down the synthesis or inhibiting the synthesis of alpha amylase, so they will not get the energy, this seed will not get energy and it will stop germinating. That is why it is called as, it is promoting the process of seed and bud dormancy. It is causing all the seeds to enter in the G0 or dormant state. That is why it is also called as dormin. Ethylene can be of two types. It either can be gaseous hormone or it can be aqueous hormone. If it is a gaseous hormone, this ethylene is known as ethylene only. But if it is an aqueous hormone, if it is in aqueous state, so it is called as ethiphone. So I hope this slide is clear to everyone. So please let me know if this is clear. Which is acting up. So now, auxin ka uses and function. So what are the functions of auxin? So auxins have a lot of functions in their body. So firstly, what happens if you cut the roots of the plants and dip in with auxin? So suppose this is a part of the plant. If you dip it in auxin, it will cause the initiation of roots. So the very first function of the auxin, it is a root initiation. So whenever you are cutting the part of the plant and you are dipping it in an auxin, that is in the plant growth media, it will cause the initiation of the root in all the stem cuttings that you have dipped in the auxin media. So it will initiate the formation of root, and it's also acting as a VD site. How VD site? Because in the previous slide I have mentioned 2,4-D is used as a VD site. It will cause the killing of all the unwanted plants that are there with the main plant. So it is acting as a VD site. The examples for this is a synthetic auxin, that is 2,4-D. What is parthenocarpy? So parthenocarpy is the formation of fruit without fertilization. So actually what happens, the seed or the fruit that is formed, it is formed by fertilization. Sperm and the ovum are fused and they will cause the production of a formation of zygote. They will turn into a seed and will result in the formation of fruits and flower. But when the flower or fruit, it is formed without fertilization, without this mechanism, it is what called as parthenocarpy. So once you will spray these auxins in the agricultural field, they will not undergo fertilization and they will give you n number of fruits to you. This is what called as parthenocarpy. So the example for the auxin that is helping in the parthenocarpy is IAA, IBA, and other synthetic auxins can also help. And the fruits that is resulting from the formation of this, from the parthenocarpy mechanism by the spraying of auxin is banana. If you are spraying this auxin in the banana agricultural field, you will get n number of bananas. This is what because of auxin. So auxin is also helping in the parthenocarpica process. It is also used to initiate the formation of flowers. So suppose if the plant is not bearing any flower, but if you are spraying any auxin here, it will cause the initiation of flowers. It will help to the formation of flower in this plant body. That is what called as flower initiation. It also helps in the prevention of abscission. Now what is abscission? Abscission is the falling of leaves. Now you have already seen that the leaves that turn yellow, it will fall off. Why it is falling off? Because it has become old. The yellow leaves are old and the green leaves, they are young leaves. So once the old leaves are formed, they will fall off, but this auxin, they will prevent the abscission of green and young leaves. They will prevent of falling off in young leaves. How they are preventing? It is just because of auxin. They are having the significant concentration of auxin in the leaves that is making it green and also making it or preventing it to fall off. And it is causing the abscission of older leaves but not the young leaves. So it is also helping in apical dominance. What is apical dominance? You can see here, if you like, this is the plant, this is the initiative of the plant has not grown yet. So now if the plant is attaining the height and it is increasing, only if the height of the plant is increasing and the sideways if they are not increasing much, this is what called as apical dominance, in which the apex of the plant only grows, but it will inhibit the sideways growth of the plant. That is what called the apex is becoming dominant. That is what called as apical dominance. The apex is becoming dominant and it is inhibiting the sideways growth of the plant. That is what called as apical dominance. So whenever you spray this auxin in the field, it will always help in the apical dominance. It will inhibit the sideways growth of the plant. And phototropism, we have already seen, if the auxin is accumulated at the tip, it will cause the plant to grow towards the more lighter side. More lighter side matlab where there is sun. This is what called as phototropism. Why this is happening? Because here the auxin is accumulated and it is causing the cell to divide and the cell to enlarge. That means auxin is also helping in cell division and cell enlargement ka process. So you can see here the light it is getting, when the plant is getting or is directly exposed to light, what will happen? The auxin will be accumulated here. It will cause the plant to bend towards the lighter side because auxin यहाँ पे ज़्यादा बना है and the cells of shady parts they are dividing more. Now coming to gibberellic acid. So gibberellic acid, the four major function is fruit development. So if a plant is there, if you provide a gibberellin in an agricultural field, it will help to cause the fruit development. It will help in the formation of a fruit in plant. So suppose if a plant is bearing less number of fruit, but if you spray gibberellic acid, it will cause the plant to bear more number of fruits. It also helps in the internode elongation. So you can see in the diagram, this is a diagram for sugarcane plant. This is one node and this is other node. And the distance between two nodes, it is called as internode. What it is called? It is called as internode. So when you spray this gibberellin in the agricultural field, it will cause the internode to elongate. And what will happen? What is the consequence of this internode elongation? It will increase the size of the plant. How? Because the internodes, they are getting increased. When it will increase, the size of the plant will automatically increase. The plant size will be more tall in comparison to the previous one. This is what it is helping in internode elongation, because gibberellic acid, when I'm talking about gibberellic acid, ये तुमको बिल्कुल से दिमाग में आना चाहिए, because I'm mainly focusing on increase in the height of the plant. Increase in the height of the plant. How height of the plant is increased? Because it is causing the internodes or the part between the two nodes to elongate or to increase. Now, sometimes vernalization. What is vernalization? Now, sometimes some parts of the plant or some parts, they will require cold temperature. Until and unless they are not getting cold temperature, they will not flower at all. So they require cold temperature that can be between 5 to 20 degrees centigrade. So they require this temperature for flowering. So you have to keep this plant at very low temperature, only then you will see a flower in the plant. But if you don't want to provide a lower temperature, and you are looking for a substitute for vernalization, that is gibberellic acid. So if you directly provide a plant to a gibberellic acid, or you expose this plant to a gibberellic acid media, you will get a flower in that. That means the plant will enter to the cell cycle and will start blooming. So gibberellic acid, it is also the substitution for the cold treatment or as well as vernalization. As a VD. And also it helps in the breaking of dormancy. How it is helping in the breaking of dormancy? Because I've already stated, like suppose this is a seed. Now this seed, it is soaked in the water. What will happen? This seed will absorb water from the surrounding. This water will cause the production of alpha amylase enzyme. Now once this alpha amylase is formed, it will cause the breaking of the amylose that is there in the starch. Where the starch is stored, starch is the main sugar that is stored in the germinating seed. It is not mobilized. When it is mobilized, once it is broken down by alpha amylase, it will break the amylose unit in the starch. They will receive the energy from that and they will start germinating. Now you will get the seedling. That is what it is helping to break the seed dormancy. The seed that was first in the dormancy stage, now dormancy has broken, now the cell has entered into the cell cycle. Now you will get the flowers and fruits in the plant body. Now coming to cytokinin. Cyto stands for cell and kinin stands for division. So what happens here? It will help in a senescence effect. Delay in senescence will come into this. Firstly, when understand, it counteracts apical dominance. Now, what is apical dominance? We have already studied in Auxin. In the case of Auxin, plant was gaining height. It was increasing vertically, but it was inhibiting sideways growth. Sideways means lateral growth. But here, if you cut the tip of the plant, so we know that tip of the plant, it is releasing auxin. When it is exposed to plant, this tip of the plant, it is releasing auxin. But when we are removing this tip, now what will happen? Tip is removed, that means auxin is not at all there. If auxin is not there, who is going to perform the apical dominance? Now the apex of the plant will not be that tall. It will not be that height as was seen in the case of apical dominance. Now what will happen? If the apical dominance will not happen, this lateral growth of the plant will happen. Why? Because this auxin, it was inhibiting the lateral growth of the plant, that is the sideways growth of the plant. Now this will happen. Sideways growth will happen. It will happen by cytokinin. So cytokinin is known to counteract apical dominance that was taken care by auxin. So can we say that auxin and cytokinin, they are antagonists to each other? They are just opposite, because auxin is promoting the phenomena of apical dominance, whereas cytokinin, it is counteracting this apical dominance and it is promoting sideways growth in the plant. It is also helping in morphogenesis. What is morphogenesis? The appearance, the outer appearance. So it will help to form all the plant of the parts, whether it is leaf, it is root, it is shoot, it is flower, or it is fruits. So suppose this is fruit or these flowers, it will be formed by when we are spraying this cytokinin to the plant body, because this cytokinin, it is known to perform the cell division. And once the cell division, because cyto stands for cell and kinin for division. So once the cell division will take place, the morphogenesis will take place in the plant and it will form all parts of the body, that can be leaves, root, shoot, anything. It also helps in the cell division and enlargement. So cytokinin, it will help in the cell division process. Also, it helps in the parthenocarpy, as we have seen in the case of gibberellin and auxin. So gibberellin, auxin, as well as cytokinin, they all are playing a role in the parthenocarpy. So for parthenocarpy, if you are getting 4 options and all options 1, 2, and 3 correct, so for all 4 options, all 3 options are correct, because auxin, cytokinin, and gibberellin, they all are playing or to promote the parthenocarpy in plants. Now, what is senescence and delay in senescence? Suppose this is an older leaf. At some point of time, it will fall off. And this young leaf, it will not fall off because this is young leaf. It is having auxin. So this is what called as senescence. When the leaf is dying, or the part of the plant when it is dying after some time, that is what called, it is undergoing senescence and it is undergoing aging. But if you want to delay this senescence, you can just apply, suppose this is a yellow leaf or older leaf, or this is a green leaf. It is not undergoing any senescence, but this leaf will fall off after some time. But you don't want to fall off this leaf. You don't want this leaf should fall off. What you will do? You will just provide the senescence from outside environment. You will apply cytokinin over the surface area or over the exposed part of the leaf. What will happen after some time? You will find this leaf turning green in color. So this phenomena, it is also called as Richmond-Lang effect, because it was firstly described by Richmond-Lang. And what he described, when we are applying cytokinin to the yellow part of the leaf or to older leaves, what will happen? It will cause the leaves to become green. And now they will not be senescent. They will not fall off, and this will be now called as a young leaf or green leaf. This is what called as Richmond-Lang effect. Now this young leaf or older leaves, that is yellow in color, will now turn into green leaves after the application of cytokinins on the top of this leaf. So this is what called as delaying in senescence. So this cytokinin will delay in senescence. Now coming to abscisic acid. What are the uses and functions of abscisic acid? This abscisic acid, it is also called as a stress hormone. Whenever there is a stress in the plant body, so whenever the plants, they are facing any kind of stress, it can be biotic, it can be abiotic stress in the environment. When they are facing a stress, they will start releasing abscisic acid. Now, what does this abscisic acid do? It will induce the abscission. It will cause all the leaves to fall down from the plants because the plants had faced adverse condition in the environment. So all the leaves and fruits, they will be shed in the surrounding. And it also causes the stomatal closing. What is stomatal closing? So whenever the stomata, suppose this is a guard cell and this stomata is closed. When the stomata is closed, stomata, it is known for transpiration process. When the transpiration process is happening, gaseous exchange of water vapor in the surrounding. When the stomata is closed, there will be no loss of water in the surrounding, because whenever stomata is opening, it is causing the water loss from the plants. So as the stress is encountered by the plant body, they will start releasing abscisic acid. This abscisic acid will soon close the stomata, and this stomata will not cause any transpiration from the plant body, and all the plant will be retained inside. It will not be lost in the surrounding. That is what it is helping in the stomatal closing process. It also causes the seed and bud to enter into the dormancy state. That is why it is also called as antagonist to gibberellic acid, where gibberellic acid is helping in seed dormancy or breaking seed dormancy. It is causing the synthesis of alpha amylase enzyme, whereas abscisic acid, it is inhibiting the synthesis of alpha amylase enzyme and causing all the seeds and bud to enter into a dormant stage. So it is called as an inhibitor of seed germination. So overall, we can say that abscisic acid, it is an antagonist to gibberellic acid. So now coming to the last phytohormone, that is ethylene. Ethylene is also a gaseous hormone. Why it is known for? It is known for ripening. So what all tomatoes and fruits ripening that you are having from the market, ripe fruits, they are just because of ethylene. Ethylene is a gaseous molecule, and it also has its aqueous form, that is what called as ethyphone. I've already discussed, it is called as ethyphone. So now let's see what are the uses and functions of ethylene. So it helps in the abscission and senescence process. Just like abscisic acid, it is also causing the flowers, leaves and fruits to senescence and abscise. It all will fall down. So it is helping in the abscission as well as senescence process. It is also helping to break the dormancy, just like gibberellic acid. So can we say this ethylene, it is helping to gibberellic acid? Yes, because it is helping to break the dormancy. So breaking of dormancy, it is regulated by GA as well as ethylene, but this dormancy, it is induced by abscisic acid. That means now abscisic acid is an antagonist to both gibberellic acid as well as ethylene, because ethylene is also helping gibberellic acid in breaking the dormancy. It induces the formation of alpha amylase enzyme. So it is helping in the ripening process. How it is helping? And one more function of ethylene is it is promoting the formation of female flowers. Why it is promoting the formation of female flower? What is the significance of this? So suppose female, it will have ovary. If it is having ovary, it will have more number of fruits. It will undergo fertilization and more number of fruits will be formed that will be ripened. So this is one of the advantage of ethylene, that is helping to promote the formation of female flowers, and this is seen in case of cucurbites. In cucurbits plants, Cucurbitaceae family, they are promoting the formation of female flower that will have ovary. This ovary will enter to the fertilization stage and will get the more number of fruits that too will be ripened. This is what the uses and function of ethylene. Ripening of fruits, it is helping to break the dormancy, and in turn it is indirectly helping to gibberellic acid by inducing the synthesis of alpha amylase enzyme. It is also helping in abscission and senescence process and also promoting the formation of female flower in cucurbites. So now we are done with plant hormones as well as the phases of plant growth. Now we'll go to questions. If you have any query, you may ask here. And now we'll proceed to questions part. We'll see how many questions and how questions are asked from this topic. Any questions and how? So no more questions. So now let's proceed and practice questions. So which plant growth regulator is an adenine derivative? So we all know that purified adenine, it is a precursor for cytokinin. So what is the answer for this question? It is cytokinin. And the examples of cytokinin are, they are kinetin, they are BAP, 6-benzyl aminopurine, as well as zeatin. So out of which one cytokinin is given, that is kinetin. So here directly cytokinin is not given, but an example for this is given, that is what called as cytokinin or kinetin. So the correct answer for this question is option number B. Name the plant growth regulator which upon spraying on sugarcane crop, we have studied sugarcane in one hormone only, increases the length of the stem, that is it is focusing on internode elongation, and thus increasing the yield of the sugarcane crop. The right answer for this question is cytokinin, gibberellin, ethylene, or abscisic acid. So this is gibberellin, because when we were spraying the gibberellin on the sugarcane crop, it was causing the internode to elongate, and this way it was helping the plant or height of the plant to increase. So the right answer for this question is gibberellin. So the right answer is option B. Which of the following is not an inhibitory substance governing seed dormancy? Not an inhibitory substance, which is not an inhibitor to seed dormancy. So we all know that for seed dormancy, the inhibitor is abscisic acid, but they are asking not an inhibitor of seed dormancy. So who is not an inhibitor of seed dormancy? It is gibberellic acid as well as ethylene. So we'll see what is there in the option. So gibberellic acid is there in the option, so we'll pick this option and the option goes right. So the next question that is here, you are given a tissue with its potential for differentiation in an artificial culture. So suppose you have tissue that is there in the artificial culture or plant tissue culture media. Which of the following pair of hormone will you add to the medium to secure shoot as well as root respectively? So how shoots and roots will be formed in the plants? How it is formed? So if it is auxin, it is promoting the root initiation, because one of the function of auxin, it is causing the root initiation in stem cutting. So for auxin, we have root. So if auxin we are providing in plant tissue culture media, that means we'll have the roots in the plants. We'll have roots. And for shoot, so now match the option. For root, we should have auxin. But we know abscisic acid is an inhibitor of everything. It is not at all initiating the root and shoot formation process. But cytokinin, it is causing shoot elongation. Why? Because the name only suggesting cell, cyto means cell, kinin means division. That means it will help in the division process and it will cause the formation of shoot. So for auxin, we have root, and for cytokinin, we have shoot. So auxin and cytokinin, they are the combination that will cause the formation of shoot as well as roots in the plants. So fruit and leaf drop at early stages can be prevented by the application of. So suppose fruit and leaf drop is happening. That means the plant is undergoing senescence. But if you want to prevent this senescence, so we have already studied a phenomena called delay in senescence. So who is delaying this senescence? Cytokinin. So here we have to answer how we can prevent this senescence, this falling of leaf. This can be prevented with the help of cytokinin hormone. Then the next question, that is question number 7, you have on the screen is, which one of the following growth regulators is also known as stress hormone? अभी मैंने बताया in the previous slide, stress hormone किसको कहा जाता है? It is abscisic acid. Why stress? Because whenever a plant is facing any kind of stress, it can be biotic, it can be abiotic. Whenever it is facing a stress, it will cause the antagonism to every kind of hormone. It will initiate the dormancy period. It will not cause the formation of root and shoot. It will cause all the leaves of the plant to fall off, all the roots, all the shoots to shed off. This is what called as stress hormone. So here the option D goes right, because abscisic acid is a stress hormone. So assertion and reason type question. Question number 8. In vernalization, flowering is induced by low temperature. Low temperature means vernalization. So we all know that flowering, it is induced by low temperature. Reason, ABA is growth inhibiting hormone. This is not the reason for this assertion, because in sentence 1, it is talking about vernalization. That means it is focusing for the substitute of vernalization, that is gibberellic acid. But in the second sentence, it is talking about abscisic acid. So the reason and assertion, they are not related to each other. But yes, we know that ABA, it is a growth inhibiting hormone, because it is also known as a stress hormone. So both the statement are correct, but reason, this is not the correct explanation for this assertion. That means the option B goes right. If both assertion and reason are true, but reason, it is not the correct explanation of assertion, because we know that ABA, it is a growth inhibiting hormone, right? And vernalization, flowering, sometimes flowering, it is induced by low temperature or vernalizing substances. But if we are providing GA in the surrounding, this vernalization will happen and the flowering will be induced. Here, the option B goes right. So cytokinins examples, we have already seen in the example slide, that is the types of cytokinin. We are knowing that there are 3 types of cytokinin: kinetin, zeaxanthin, and BAP, that is 6-benzyl aminopurine. So the option A goes right for this. So you have a couple of questions for your assignment. So just go through it and try to attempt. If you have understood all the things, it will be very easy to make these 2 questions. So here you have 2 questions for your work. So thank you everyone for watching this session. If you like the session, do like, share, and subscribe. Also, you will get the freebie notes in the form of, like, you will have all the important topics list that you can study for your NEET exam. Whether it can be physics, chemistry, and biology, just visit to that link and you will find the important topics for all the subjects that will be there in the NEET exams. Thank you everyone for your time and patience. Meet you in the next video. Till then, bye-bye. Take care. Wish you all the best.